this workshop here, and as I will say several times, we are very excited about this new generation of workshop that we are putting together. So this workshop we are going to focus on enhanced sampling and free energy applications of biomolecular modeling and simulations, which is really the next generation. Once you have learned this simulation, this is really the next level of quality and sort of techniques that we can apply to understand systems better. My name is Emad Tajkhorshid. I'm one of the faculty members and director of NIH Center for Macromolecular Modeling and Bioinformatics that is sponsoring the workshop. We have been giving this workshop for more than a decade now. And I will just very briefly introduce the center, what we do here, maybe a few slides, and then talk about some of the applications, just motivating why you would like to know what will be presented in the workshop this week. Very briefly, I try to make it brief because you have a lot of exciting instructors and exciting material to learn about. Yes, I mean, this is, again, the first workshop of this kind we are putting together. The entire week, we're going to concentrate on enhanced sampling and free energy calculations here. Back to the center that is supporting, so many of you might know of these two programs, NAMD and VMD, a simulation program that you will use a lot, and a visualization program that goes hand in hand, or a sister program of NAMD. So these are have been produced by this center, the P41 Center, supported by NIH for more than 25 years now. And these are the main products of the center. And as you can see, the number of papers, the number of research sort of applications applying MD has been growing kind of exponentially. You can see how the number of papers have been increasing. And we are providing the technology in the center. We are developing the program, examples, also train people how to use the program, et cetera, et cetera. By very conservative measures, we have more than 100,000 users for VMD. That's more popular because you can use it with any MD engine that is your favorite, any molecular system essentially. And then NAMD users are about 20,000 users. But NAMD is #1 program used at many supercomputing centers. Not just MD, because any application, any supercomputing application, NAMD has been taking the largest number of cycles at NCSA, for example, or Oak Ridge National Lab, etc. These are the number of citations that the papers written for NAMD and VMD are receiving these citations. So you can see, like for the case of the VMD is more than 2,000. So we calculated almost every 2 hours we're getting a citation. Somebody is using and citing the papers, which means that this program. So we feel really committed to this very large and growing community. And that's what we do at the center. So now we do our own science, like yourself. We are interested in doing research. But on top of that, we are sort of focusing to make the program available on all platforms, for example, that come online. Now we are working with people who are working on the next generation's hardware coming online, and in the next Blue Waters or at Oak Ridge National Lab or at Urbana National Lab. We are working with the vendors even actually to make sure that the program is good and works fine, and it sort of scales very well. We try to actually consistently add user-requested features to this program, both in terms of simulation, analysis. You will see some example, visualization, especially for these sort of more complex kind of simulations that we are planning to do. And there are more seats here, if you guys want to. And then we are covering a range of applications going. Actually, you can visualize orbitals in VMD these days, all the way to cellular scale phenomena. And I think one thing I would like to emphasize is that we spend a lot of time to make this feature accessible to a wider community of users. Maybe you just want to use some free energy calculation for a few weeks and be done with it. You don't have time to really learn all the nitty-gritties. So we try to make it as easy as possible by providing actually a lot of additional tools. On the hardware technology, I pointed out that's extremely important to have the program fast. That's one of the reasons you are here. NAMD is probably the best sort of parallelized code on very large number of processors. That has been the emphasis in NAMD. So we are working with Nvidia, in terms of GPU porting of the code, And you can see we have very close collaboration. We have actually one computer science faculty affiliated with the center from day one, who has been only working on parallelization of the code with multiple students. So this is really a strong aspect. You can see Jim Phillips and John Stone and Antti-Pekka, who was unfortunately passed away. He was actually somebody at Oak Ridge National Lab joining Nvidia, working on NAMD just to make sure that the code runs better and more efficient on GPUs. A couple of examples of what I pointed out in terms of making the tools and applications methodology accessible. So this is what we call interactive MDFF, in which we allow experimentalists, for example, when they want to fit their structures into densities in an interactive manner without really learning all the commands and everything, so they can actually use this interface and fit their structure. While an MD engine is working behind the scenes, they can visualize the system and drag the protein sort of a structural element that you would like to fit into that density while you're monitoring, for example, the quality of your food. This kind of interactive sort of interfaces we would like to do. So there is another example, Quick MD, as the name suggests. This is there to just allow you to go through all the steps involved in setting up a typical MD simulation, taking the protein, cleaning it up, putting it in a solvent, taking care of titration states to some degree, and even actually putting it in a membrane, et cetera, and we are actually expanding on this as a sort of as a tool in which you can even have access to some free energy calculations, sort of setup, also analysis, etc. We would like to run an Amazon. Maybe you don't even want to bother with setting up a computer at home, so you just want to work remotely on the cloud. So we are working on all these fronts really to make the program and the technology available and accessible to people. In terms of training people, we have been doing this for a long time. This number might not be accurate. This is a few months ago. We have had a lot of workshops. Many researchers have been trained from a high school level all the way to people who are working in industry, faculty, postdocs, lots of students, obviously. And a lot of material that sort of exploded over the last few weeks because of this workshop, and you will see are available on our website. So I really encourage you to check. You will find a lot of useful material there at different levels. I guess all of us are interested in doing simulations. That was a brief introduction of the center and what we do and what we are committed to and our commitment to the users. But let me now switch to sort of introducing you to why we need enhanced sampling technology. And as I was saying, so many of us are interested in understanding molecular phenomena, or biological phenomena, or any phenomena at the molecular scale or atomic scale. And in that regard, simulations and the simulations are extremely powerful. Sometimes the phenomena you're interested in happens on its own natural time scale because you are not able to run sub-microsecond simulations rather routinely. So you just just watch the system, put a ligand in solution and see how it binds, for example, in this case, to its carrier, ADP being carried. And then you can analyze the system, what happens in terms of electrostatic interaction between these things, et cetera, et cetera. So these are areas in which you can take advantage of this microscopic view or detailed view that you get from a molecular system. Here's another example in which the group of D. Shah looked at binding of a drug to a receptor, a GPCR, and this took several microseconds, sort of on the microsecond time scale. You can see now how the drug explores the protein, hits it a few times before it really finds its binding pocket where it sits for a rather long time. Okay, I mean, see, if you can manage to run microsecond simulation, a few microseconds, then you might be able to see spontaneous binding of a single ligand in your simulation system without doing anything. But these examples are rather limited. So we have only maybe very limited number of biological phenomena that happen on a microsecond. Most of them happen millisecond or longer. So we have to do something about that. Here's another example in which you might be interested in sort of in terms of nanobiotechnology, there is a molecular model that they have put together instead of having a cartoon showing how antibodies might be arranged on a surface of a substrate capturing viruses, so we can imagine why you might be interested in a more microscopic and more accurate description and more realistic representation of the molecular phenomena. And here's another example. Again, you can see in this case we are looking at a recent publication from the center in which we looked at ion permeation through a channel and without inclusion of the lipids that you can see nicely showing here, dancing and forming actually part of the permeation pathway. It was impossible to understand the behavior of this channel because this channel was completely closed at the bottom and they couldn't figure out what is the permeation pathway. Only when you package the lipids, you see how the lipids contribute and line the permeation pathway. So these microscopic views, these are things that we cannot yet experiment with. You can do it, normal simulation, good. Not very exciting, I would say. It's nice to observe them, but that's routine, but many of them don't take place. So that brings us to, you always have issues with the time scale of the simulation. You never can run it long enough to capture it completely. Therefore, you go to sort of realm of non-equilibrium simulation. You have to do something to the system. And of course, you want to quantify the process using free energy calculation, in which you need to make sure that you have a sufficient sampling to have rather accurate description of the process. So that's the whole idea of this. So let me give you some example. The first sort of kind of generation of attempts that people did in terms of being able to simulate or to observe something that might even resemble biological phenomena was sort of including additional forces into the molecular system. For example, you can say the steered MD was one flavor of this kind of application in which you steer a molecular system into a particular direction, and you can be very clever about the application of these forces, combine different magnitudes, different directions of forces applied to different groups of atoms to create. There are a couple of spaces, sit here if you want. So you can, for example, you can sort of pull a substrate out of the binding pocket. You can look at an ion permeating the channel. You can even apply torque if you just apply tangential forces to a set of groups of atoms, etc. You can create a gradient across the membrane by pulling everything down and things of that sort. But then, of course, this allow you to induce a phenomenon in your molecular system. But then, of course, at the end, you want to be able to somehow connect this at the end of the day to the free energy, which is the real quantitative metric for you. So this is one example, a very old slide in which we are pulling a substrate through a channel, and we can have different schemes applying constant force or doing it a variable force or what we call constant velocity, sort of increasing the force applied to the substrate to slowly pull it. And sort of in an adaptive manner work with the force so you don't apply a large force. And then again, there are, for different methodology, for this kind of methodology in which you are, for each simulation, you're always in a non-equilibrium regime, so you can actually, there has been some formalism allowing you to convert your, the work that you're doing to the system, which is always, on the average, larger than your free energy. There has been formalism developed showing that actually the exponential of the work, if you look at the average of that, then you can get the free energy of the system alone. Of course, if you have enough sampling of this little rare events that happen at sort of at this scale of your distribution. So again, the point is not here to emphasize, but again, you see this is one flavor of applying non-equilibrium methods and trying to, at the end of the day, doing free energy, getting free energies out of them. Sampling is always the keyboard here. So for some reason, nature spends a millisecond to do something, and now we want to do it in a few, I don't know, in a microsecond or a few hundred nanoseconds. So there is always some limited sampling in this problem. But then the idea is that maybe there are a lot of unimportant, random events happening in a molecular simulation or a molecular system that we can sort of avoid by cleverly sort of selecting the specific pathway, reaction coordinate, etc. Let's skip this one. So then you can get free energy profiles and compare, for example, two channels. Why one channel doesn't permeate a particular substrate, whereas the red one is something that allows the substrate to go. Another class of examples in which we have been applying these enhanced sampling techniques and non-equilibrium simulation have been large-scale conformational changes in proteins. This is just one example in which transporters have to undergo very large conformational changes to do their job. And here you have to really design the reaction coordinate much better than just RMSD. You go into the realm of collective variables and how to combine these things, and we have one day dedicated to this very important and fast-growing aspect actually. So you see we are interested in this very complex process. I mean, if you do RMSD, you completely mess it up. If you use RMSD as a collective variable or your reaction coordinate, it's impossible to capture this complex sequence of events, let me say. So we had to, these are some examples of different transporters all doing the same thing, taking something from one side and delivering it to the other side, but you can see their modes of conformational changes are completely different. Depending on the source of energy, for example, this is ATP coupled, this is ion coupled, and this is a simple antiporter rocking back and forth. The point being, I mean, these are very complex molecular conformational changes, and in order to handle them in a way that you don't have to sample the entire space for milliseconds or longer, you have to really find the most optimal reaction coordinate. So for this, actually, again, Mahmoud, who will be presenting the last day, he will talk about how to select a little bit more systematically, how to sample the configuration space a little bit better. And then, for example, instead of just going linearly using RMSD, you can imagine these are your two end states, and you would like to describe the process. You can take a simple reaction coordinate, do a targeted MD in which you reduce the RMSD toward the final state, your target state, in a time-dependent manner, how much time you can spend. But if your actual transition pathway, the real one that happens naturally, is too far, then you will have no chance, essentially, to get there, even if you apply all the path refinement algorithms and techniques that we have. So we have to explore a little bit better here. And again, so based on defining different collective variables, you might be able to steer the system, not just through RMSD, but through combination of helical orientations or distances and rotations and things like that. So, and when you do that, you will see that you always improve the quality of your initial guess. Instead of using this red guy, you might have something like this that has a better chance to converge to your real transition pathway eventually. Again, I pointed out, so the idea is sort of having a time-dependent collective variable and changing it from the initial state to the final state in a clever way. And these are some of the collective variables that are already available, implemented in AMD by people who will be actually teaching you this later during the week. And you can mix and match them, combine them, have a linear combination of many of these things to induce the most optimal mechanism that you would like. And then when you do that, then of course you can calculate the work and see how difficult it is. This is something that you will see more in the last stage, how difficult it is to go through this pathway or this pathway. These are all very quick simulation, 20 nanoseconds. But if you design and work with these collective variables, then you can see actually how nicely we can make it easier for the protein to undergo that kind of conformation change and things. Then you can take those and actually calculate free energies. So this is, for example, a mechanism that we induce, and we can see how nicely you can even actually do it in a complex environment of the membrane, etc. So then again, this is just one more example in which we use there's combination of a collective variable to many collective variables, some of them dealing with the substrate binding, unbinding. So this is a protein that binds the substrate, flips to the other side, delivers the substrate, becomes APO, and flips back. So you have 4 legs in this thermodynamic cycle that we constructed, and you can look at some collective variables describing your substrate bindings. This is something that you will hear more today and tomorrow. And then you can have other collective variables describing your protein conformational changes. And then at the end, when you sample these legs, then you can take many replicas representing this cycle and put them all in what we call replica exchange kind of simulations. And that's something else that you will hear more this week about. And do one simulation that allows you to get the free energy profile associated with these 2 pathways. Nicely showing that without the substrate it's very difficult. You have a high barrier. With the substrate, your free energy profile changes. You have numbers here matching exactly what the experimentalists sort of suggested. This should be something like that, of course, without any numbers back then to this. But now we have the complete profile and what happens and everything. So this is these are some examples. So let's see, this is another example in which we are trying to look at, let's say, during the vesicle fusion to the membrane and neurotransmitter release from a neuron, you have a very large sort of structural transition of your membrane system. I mean, something that might not be even possible to do using collective variables. So now for this even we have actually methodology in place that allows you to induce what we call shape transformation using the multi-scale approach, in which you apply some grid potential and sort of in a time-dependent manner you change the shape of the grid that controls the behavior of the molecular species on top of the force field calculations. So by the way, the assumption is that all of you are expert in running MD simulations, you know what it is, and you know how to set that, so we are going to the advanced mode. So if that's not the case, you might want to talk with one of us and see what we might be able to do. So this is, for example, some examples in which we are inducing, you see, again, using this multi-scale, non-equilibrium approach in which we are adding additional constraints or steering the system toward a particular direction. We are inducing a sort of a shape transformation. This is a simple case, kind of sine or cosine wave you can induce. I can change this, change the phase of this during my simulation in a time-dependent manner and induce a continuous shape change in the system. Again, this wouldn't happen, of course, if you just do normal MD simulations. So unless, I don't know, maybe you can run a millisecond simulation to see sort of a large or very large simulation using coarse grain in which you start seeing undulation and things like that. So another application of this sort of advanced technologies in which we are steering the system into a particular direction is when you are trying to put together models that are at the level or at the scale of cellular systems. So, I mean, just putting them together requires a lot of effort. One famous application is what we call molecular dynamics flexible fitting. Again, so electron microscopists usually come up with a low resolution electron density of a molecular system, and you would like to build a structural model into those densities, right? So especially nowadays, DM has reached resolutions of, I don't know, 3.5 angstrom or so, so you can reach very high resolution using single particle DM. So the idea of how to build the structures into their density, and I don't know if anybody has done this before, but essentially they are doing it by hand, grabbing these helices, using tedious programs to fit them into those densities, build a model, and then put them there. So what we are suggesting or we are offering is now we can do MD on the molecular system. You build an initial model, or you take it from a different conformation of the same system in a different state. You start with that, and then you slowly put it into that density. And in order to do that, then we can see we can define, introduce that density as an additional potential to your simulation. and then guide and steer and ask all those atoms to be attracted and gravitating toward those densities. And by that, you can get actually a molecular conformation, a conformation that also sort of follows the natural bound angles and bound distances, sort of in a natural, flexible manner, you fit your molecule into the new density. It is another example in which actually you can work with the different kind of levels of this density, initially having a very low resolution, because if your density is high resolution, then your molecular system gets stuck into the rugged landscape there. So they smooth it using simulation trick to allow the system to fall in there and then slowly increase the resolution of the density to be able to have the best fit. Again, these are all kind of, let's say, outside the equilibrium realm. So you have to add additional constraint, additional potential, additional forces to be able to make the system to visit parts of the configurations. Okay, so then these are the last examples. I try to minimize my time because you will have much better stuff coming up. And this is the last example in which we are actually trying to put together something that we call, let's say, the sort of cellular envelope. So like how to place a number of membranes, a number of lipid molecules into a particular shape. We start with a sphere, and then we actually convert our protein, for example, to a sort of very coarse-grained representation in which we distinguish between the membrane part and the soluble parts of the protein. Then we use this reduced representation. That's another way of increasing your sampling power. This reduced representations of each protein and throw them into a density, a sort of gravitational field that represents our membrane. You can see you can have hundreds or thousands of these proteins floating around. So once you have this initial randomized configuration of your protein, then you can convert them to real molecules and then use their densities to carve out perfectly a lipid sort of hole that allows the placement of the protein. Again, I mean, the point here is that without using this bias simulation or non-equilibrium simulations in which we are guiding the system toward a particular way, you won't be able to even construct the initial model for such large molecular systems. So this is just to give you an idea. This guy has 1,400 proteins and 100 million coarse-grained particles. We have run very short simulation on a billion atom system in NAMD. It's not released yet. We still have to work on it, but that's another sort of front in which I think NAMD is unrivaled right now. So, but anyway, so this is just to put this together. Everything should be automated, etc., etc. So I'm going to skip the next example. This is another example in which we are sort of and go to this last example in which we are using, again, biosimulation to construct a nanoparticle, gold nanoparticle. We want to cover it with lipids, and that's what we are doing. So this is the experimental picture. They use this cartoon to optimize their design. So obviously you draw this cartoon and then use it. Whereas we would like to be able to do it using simulation. So we cover it with a particular chemical group, and then you can even simulate. And then at the end, what we are going to do, we're going to add lipids surrounding it and allow the lipids actually, again, applying some biasing potential or gravitational field. We let the lipids to land on this nanoparticle. It's a collaboration between an experimental lab doing very exciting work on these gold nanoparticles to deliver for drug delivery, etc. So now you can see actually we are going to add the lipids, and again, by using sort of in a time-dependent fashion, using some potential, we guide the lipids and invite them to sort of sit on the nanoparticle, and that's the way to. So lots of applications for when you get out of the equilibrium, regular, and there are lots of applications for this, and hopefully we can actually teach you some of these techniques. So as I pointed out, this is the first workshop in which the entire week is dedicated to these non-equilibrium techniques, enhanced sampling techniques, and free energy techniques. We have a great collection of instructors, really the best people in the field, so you will see that. So many of them have implemented some of the features that you will hear in NAMD, have been working on it for a long time. A great program, so going alchemical free energy calculation, transition path sampling methods. Chris Chippo will be covering these two. And then we have Brian Braddock, who will cover constant pH simulation, for which we have a brand new tutorial. How many hours is the age of this tutorial? A couple of tutorials actually were because just this morning, and so they might not be even linked. So people really worked very hard to develop this, because this is a new type of workshop we are doing. If you have any feedback, please let us know. That would help us improve this for the next workshop that we will do. Geometrical transformation and collective variables. This is something when you are dealing with complex molecular systems, you have no way to stay out of that. You have to learn that, or you're stuck with just RMSD, which is not very insightful in terms of what is happening. So a special algorithm for enhanced ergodic sampling. Again, Chris is going to cover this day. And then at the end, we have reaction pathways, somehow application of these collective variables to complex reactions. So a lot of material and a lot of fun. I'm so excited about this workshop. I invited all of my own students to come and listen to the talks. We have a lot of additional people from the campus who are interested in that.